Morning, everyone. So Gabby there was presenting from a kind of ethical point of view. I'm going to present from a slightly different perspective, thinking about actually from a practical scientific point of view, thinking about these issues as how they affect the validity of our science, or try to demonstrate this. Okay. So first I'll just give a bit of context, and then I'm going to come into some discussion of scientific sustainability, then try and link that in with the environmental sustainability, and then give some ideas about what we might want to do in order to solve some of these problems. And I just want to emphasize that I'm going to present some empirical results today that are mine, but I want to emphasize that for all the problems that I'm talking about, there's wonderful projects going on and people doing amazing work that I'm not going to mention today, and I just want to highlight that people can go and look for that stuff, and if you're interested. Okay, so at the moment I'm working in Taipei in Taiwan, but I'm not originally from there. So originally I'm from Scotland, although I'm more from this type of Scotland rather than this one, but we'll make the point with the nicer picture. So as an academic, and I'm sure you're all familiar with this, you end up living lots of different places around the world and constantly moving. And in that process, you observe just how variable people are. So the human species, as I'd like to say, is a really weird animal. So we live in all the environments on the planet, which no other animal does. We live at sea level, we live up at 4,000 meters, we live in the jungle, we live in castles sometimes. We form bizarre social structures and relationships. Really bizarre if you're Scottish and they keep losing in the football, but there we go. And also we make really bizarre noises and artwork and so on, which no other animal on the planet does. And I use these examples of the weirdness of humans from my own country, because I want to really emphasize that, to everybody else, everyone's weird, which is great, and I think that's wonderful. But as scientists, especially scientists who are trying to study the human brain, as I think we all are, since we're at the Human Brain Mapping conference, to do that, we need to understand the brains of all the humans, or all the types of humans, I would argue. Now, there's exceptions to that, of course, that if we're looking at really specific subpopulations, like disorders or age groups and so on, you can argue that you don't need to be so widespread in your research. But even there, I would say that even if you're studying a specific disorder, you're likely to find interactions between the environment that a person lives in and the manifestation of the disorder that they might be experiencing. So I think we can generalize safely the statement that understanding the human brain means understanding it in all of its variety. So to go back to first year stats class, when we're doing our experiments, on the whole, the structure of our scientific philosophy that we're adopting is this sort of frequentist approach, where the idea is we have a population, we want to understand the population or compare between different populations. And so what we do is we randomly sample from that population to get a sufficiently representative subsample that can represent that population for us. And we get beautiful Gaussian distributions all the time, of course. The problem arises, though, if we're not randomly sampling from the population and that we actually are sampling in a biased fashion from within that population. We can still get some lovely normal distributions, but the distributions are misleading in that they are not actually representative of the population that they are meant to come from. hopefully this is familiar to everyone, and hopefully it's familiar to my students when I try and teach them this stuff in stats. So the question is, how well are we actually sampling from the human population? And this was a question that I did some empirical work looking at this question in the context of neuroimaging to try and see just how well we are actually sampling humans. Now, I think we all have suspicions about what the answer is to that is going to be, but as scientists, it's our responsibility to not just have suspicions, but to go and actually study them and try and prove them to be true or not. So what we did was to get about 120,000 neuroimaging publications from a variety of journals, trying to sample across prestige levels in journals because of the biases inherent within access to those prestigious journals in terms of geographical location. We then went through and just used the metadata to get where these papers the authors were coming from. And then we used the abstracts and the titles and the keywords attached to them to classify them as what imaging modality that they were using. So MRI, PET, EEG, and so on. And finally, once we had this information, we could then relate that to the economic metrics of the countries that the publications were coming from. So this is a map of that outcome, showing the distribution of the sample of neuroimaging publications around the world. So the color bar here goes from one publication over the time period that we sampled, which was 10 years, up to 15,000 publications. So countries such as the United States are at the upper end, where we were getting about 15,000 publications. And you can see the countries that are closer to a sort of almost black color, where there was one in the sample. And what we can see here is that primarily what we're doing is sampling from North America, Europe, and increasingly China. But specifically, I would say within China, a specific subpopulation within China, which is the Han majority within Tier 1, Tier 2 cities, and generally excluding minorities and those who are living in other areas. So what our sample actually looks like is this, although considerably younger, I will say. So we're getting, as I say, people North America, Europe, and China. So what we are doing, I would argue, is exactly the thing that we should not be doing if we want to understand the human brain, which is producing a biased sample that we then make general conclusions from. The issue goes beyond just the locations that we're sampling from, but we actually see biases within the type of neuroimaging that is being done in different places. So we can see that MRI is more common in some places, EEG predominates in others, and so on and so forth. So what this would mean is that whilst we might be sampling reasonably well from some population, we might only be getting specific types of information about that population. And so if we're making conclusions about, say, structure, where we maybe want to use MRI, we might have wonderful information about structure from a particular place and none from another. If we're looking more EEG, we might have okay samples from some places, but absolutely nothing about the structure of the brains in those places, which I'm sure we can agree would be less than ideal when we're trying to put together a coherent picture of the brain as a whole and not just little aspects of it. So the conclusions I think we can take from these results is that indeed the human brain is currently very poorly sampled, which means that we're probably missing out on important variation in how the brain operates in different environments. So how does the cerebrovascular reactivity manifest itself when you live at 4,000 meters compared to when you live at sea level? It's an interesting question. I have no idea because no one's ever looked at it. And we can think of so many different questions where our environmental context, we can imagine, would produce really interesting variation in the fundamental biology of the human brain. We can also conclude that the types of information that we're getting about human brains is biased based upon the locations that people are being, I don't want to say experimented upon, studied at any given time. And I would argue that this is limiting our knowledge of the human brain. So, what is it that causes this biased sampling? Again, I think we can all have suspicions about what the answer to that question is going to be, but we did the correlation, so we've proven it. And the answer is, of course, money. So what this plot shows is the number of publications from a given country against the GDP of that country from the World Bank information and from the Taiwan government information. And it's not just the amount of money, but it's the amount of money that's available for research. So we can understand that different countries, based upon their socio-economic conditions, are going to have different priorities in how they allocate money. And this relationship is almost suspiciously perfect for someone who looks at graphs. But interestingly, the economic resources available are not just determining how much research is done, but also what type of research is being done. So what this plot is showing is the proportion of articles from each country that uses a particular modality. And in the blue line, you can see that the proportion of work that's being done with MRI increases as the country has more financial resources available. And the amount of research that's being done with EEG goes in the opposite direction, where countries that have fewer resources available are doing considerably more proportionally EEG research compared to MRI specifically. The other modalities, there was no clear relationships, but they were much less well represented in the sample because people don't do as much PET and intracranial recordings and so on. So the overall conclusions I would maybe make from these results is that our current inferences about the human brain are potentially invalid because we have not appropriately sampled from the population that we're claiming to study. And that we can also see that this potential invalidity in our inferences is stemming from the economic realities of the global system. And I would argue that continuing to just do what we're doing and to continuously produce these biased samples is not scientifically sustainable because it means that we will not actually achieve the aim that we claim to be setting out to achieve, which is to understand the human brain. So what's the answer? So do we just give MRIs to everybody? Maybe, and we probably should be to some extent from a medical point of view, but from a research point of view, I don't think this answer of just doing more is sustainable, bringing that into the environmental sustainability issue. So as we do more and more and more research and try and produce more and more research equipment and do more and more research analysis, we are causing these environmental issues of environmental degradation and of carbon pollution. So as we are making more and more research equipment, all that equipment is built on resources that are extracted, usually from the countries that are not being actually studied with the equipment that we're manufacturing, which is causing environmental degradation, terrible human rights abuses, and so on. At the same time, as we're doing these analyses of bigger and bigger samples, or even just doing our own little analyses on our 20 subjects, we're producing carbon pollution from the power that we are using to do so. So I would argue that trying to answer this sampling issue through just a massive expansion of research would be environmentally unsustainable. And so I'd like to put forward a few little ideas about solutions. I don't think I'm going to hit upon the right answer to this, so these are just some ideas I'm going to put out, and it'd be wonderful to hear other people's ideas and opinions. But these ideas, I think there's a lot of overlap, and the previous speaker kind of hit on this as well, of an analogy between the global scientific system and the global manufacturing system in terms of e-waste. So within the current global manufacturing system, we produce massive amounts of this waste because the structure is a sort of linear structure. One must produce more and more and more to be able to obtain the resources that one needs to keep doing what one does. You produce something, you throw it away, you make something new, you throw it away, you make something new, and so on. So this is a infographic sort of thing here from the Royal Society of Chemistry, who have done some work on e-waste and put forward some ideas of how we can transition from this wasteful structure to a more sustainable structure. And I'm going to highlight a few parts of that, starting with the idea of reuse of resources. So if we want to be studying underrepresented populations, we may want to distribute existing imaging resources to those populations. Importantly, I don't mean that in a sort of charity sense, in that we take our old EEGs and then just dump them in South Africa. What I mean is that we use these excess resources as opportunities to start building up relationships. So many labs in rich countries and rich institutions do have extra resources lying around that just get left to gather dust. If these could be seen as an opportunity to form a connection with people in an underrepresented country in a long-term partnership, I think we could develop an improvement in the situation in terms of our sampling, as well as other benefits. But I would just emphasize again that this couldn't be done in a sort of charitable sense of coming in and just giving people old junk. It has to be this partnership style, I would argue, where we continuously work with people to share technical and practical knowledge. The second part of this structure would be to reduce the amount of resources that we're using by potentially reducing the amount of research that we're doing, but focusing it in a more appropriate fashion. So the way that money is allocated within countries that have a lot of money to allocate to research also follows this kind of linear do more and more to get more and more back structure. And I would argue that this structure produces unwanted consequences, unwanted both for the people within those countries and globally. And so we might want to consider how we want to restructure our scientific endeavor in a more sharing sense rather than competitive. And that fits in with this idea of rethinking and redesigning structures. As our previous speaker said, individual solutions to these problems are unrealistic and often counterproductive when the problems stem from the structures that we operate within. So, as I've said, the current research structures that we all work within, or I assume that we all work within, follow this competitive model. And, well, if you go around the conference and listen to what people are talking about, everyone's complaining about it. So everyone knows the problems it produces, apart from a select subset of people. So there's probably energy within the community to actually change this, potentially. And we can take inspiration, perhaps, from people who have analyzed and criticized the economic structures that we operate within that I think are highly analogous to the research structure that we have developed globally over the past decades. In that they are both unhelpful to the people participating, but also necessarily produce a lot of waste and environmental degradation. So we might want to reconsider these structures, as I say, along non-hierarchical and associative models rather than hierarchical and competitive ones. So as a final conclusion, I would say that we're faced with these intertwined challenges. So these environmental challenges that we're very aware of, I think are inherently interconnected with the challenges that we face from the validity of the science that we're trying to do. And that these two things can't really be separated from each other, which is both a daunting thing, but I think it can also be quite an inspiring thing, in that as we work to solve some of these problems, we can also be helping to solve the other ones for no added cost, which is nice and sustainable. So I think, in conclusion, that it would be productive for us to start thinking about these structures and talking about ways that we can maybe adapt and modify them to improve both the scientific problems that I would argue we face and the environmental problems that we definitely face. Thank you.